MutationForecaster is a collection of tools which allows researchers to quickly examine millions of genomic variants. These tools are accessed through a web browser. To begin an analysis, upload a VCF file containing your genomic variants. VCF format is used as input to all our web tools. Optionally, a BAM file can be uploaded and used to validate Shannon Pipeline results in Vertical. Some specifics of these tools will be discussed later in the video. Additionally, individual videos are available providing an in-depth look at each tool. To the left of your screen, you'll find a sidebar with all uploaded files and the results from your analyses. Using the three buttons located on the upper right of each box, items can be viewed, edited, or deleted. The Shannon Pipeline is a tool which examines variants in coding and non-coding regions affecting splice sites. It examines variants on a genome scale and can examine up to millions of variants in a single execution. It provides a list of variants that are predicted to have detrimental effect on splicing. Filters can be selected from the submission screen. Below the filters is an optional patient identifier field. This identifier can be used to identify the Shannon Pipeline run once it has been exported to your personalized database. If no identifier is specified, identifiers from the ID field in your VCF file are used. The patient identifier field is present in all other genome scale tools on MutationForecaster as well. Results can be directly downloaded or viewed online in tabular format, scatter plots, or through our seamless connection to the UCSC genome browser. Hacia provides an in-depth look at single variants. It produces very similar output to the Shannon Pipeline, however, it also provides additional details about the variant, including transcription factor binding sites, phenotype prediction, and exon definition. You can examine variants found to be of interest after a Shannon Pipeline run, or any other variant you specify. Variants in Shannon Pipeline results can be directly submitted to Hacia through a button in the results table. Users of Hacia before its integration into MutationForecaster will be familiar with its results. Tabular data is generated for all selected binding site types. By default, all available binding site types are selected. The Phenotype Prediction tab contains data for all prospective isoforms, including the total RI across all exons, a visual representation of potential isoform structures, and a 3D graph of the relative abundance of isoforms before and after the contribution of the variant. Custom bed graph tracks are also generated and can be viewed in the UCSC genome browser without leaving MutationForecaster. VEP, a tool created by Ensemble, predicts the effects of variants on genes and protein sequences. SIFT and polyphen scores are created for each variant. Although this tool is freely available through Ensemble, we've included it to supplement our expertise in mRNA splicing variation. For additional information on VEP, please check the VEP website hosted by Ensemble. Vertical allows you to validate predictions made by the Shannon Pipeline by comparing Shannon Pipeline output to RNA-seq data contained in your uploaded BAM file. Reads related to splicing are compared in disease and control samples to compute a p-value. Full Vertical results can be downloaded or viewed in a formatted table online. Each row of the table provides an overview of a variant. For more information on a particular variant, each row can be expanded by clicking the plus sign on the left side of the row. In this expanded area, specific read counts can be found for each type of evidence, and data for each control can be examined. Cytogenetic DST is our visual analytics decision support tool. Right now, it shows copy number and other deleterious genome changes from public databases as genome browser tracks. It will soon include tracks containing significant gene variants generated by Shannon Pipeline, Variant Effect Predictor, and Vertical. The browser also displays the distribution of our proprietary probe products used for library enrichment in the next generation sequencing and for fluorescent in situ hybridization that are available for purchase from Cytogenomics. Results from any of the tools previously mentioned can be exported to your personal CUVD database. Variants can be searched for in external databases such as ClinVar, dbSNP, and locus-specific LOVDs. Other databases will continue to be added in the future. Your CVD is a place to store variants of interest found while using MutationForecaster, determine whether you have encountered a specific variant in past analyses, and discover what is known about variants in external databases. Because each variant can be associated with data from all our web tools, columns can be hidden individually using a button we added to the standard LOVD layout. MutationForecaster is your one-stop shop for variant analysis on a genome scale. It keeps your results from the variety of tools in one place, allowing for ease of use and interoperability between tools. For more information on any of our tools, including peer-reviewed publications and tutorial videos, please visit MutationForecaster's Learn More page.